got his PhD from Cambridge University. After that, he was a postdoc at KITP at the University of California in Santa Barbara. In 1985, he joined the faculty here at the University of Illinois. He is now the Swanlund Professor of Physics. He's the director of the NASA Astrobiological Institute for Universal Biology. And he also leads, he's also the leader of the Genomic Institute here at the University of Illinois. Oh, the biocomplexity group in the Genomic Institute here at the University of Illinois. So, and he's won too many awards to name them all, so I won't. But today he's going to tell us about what theoretical physics can tell us about the evolution of early life.
So thank you for inviting me to talk at this event. You know, I've given a colloquium at Harvard and MIT and Fermilab and all sorts of other places, and I'm more nervous about giving this talk than any of the other talks. So let's suppose that you have a wristwatch and you want to figure out how it works. So here's one approach. So what you can do is you can take the wristwatch, put it on the table, get a hammer, I don't have a hammer, and smash the wristwatch, and all the pieces will go flying out. Okay? That's kind of like what high-energy physicists do. Actually, high-energy physicists are a bit smarter than that. What they do is they would take two wristwatches and smash them together, and the pieces would fly out, and you get more energy that way. So that's high-energy physics. It asks, how do I, if I want to understand something, find out what it's made of? There's another school of thought that says that's not enough. If you want to find out how something got to be the way it is, it's not very useful just to find out what's inside it. You need to know how the different parts work together to explain the richness and the complexity of the phenomena that you're interested in. That approach is really statistical physics or part of condensed matter physics, and I'm going to be talking today about both types of question. The first kind of question is kind of fundamental. It's one of the sorts of questions that everybody asks. You know, how are we here? How did the universe begin? Why does life exist? All of those sorts of very fundamental, almost religious questions. And then the other kind of questions, the ones that come from condensed matter theory, are typically associated with understanding complex materials and complex phenomena, such as solid-state physics, or superconductivity, or other types of interesting phenomena where all the parts interact in some way so that the whole is greater than the sum of its parts. What I'm going to tell you is how, using the techniques from condensed matter theory, from statistical physics, from this collective view of the world, one can understand one of the most fundamental questions that we have in science, which is the evolution of early life. So I'm going to talk about that, and I'm going to include material that came from some of my students and associates who are listed here. I particularly want to mention Carl Woese, who was a professor here at the University of Illinois, and you'll hear about his work during the course of this lecture. Okay, so before we understand what theoretical physics can tell us about early life, let's ask a simpler question. What can theoretical physics tell us about anything at all? Okay, in fact, actually, what is theoretical physics? Well, that's a difficult problem, and I was scratching my head trying to think of how I would explain that to you, but luckily this week the Nobel Prize Committee came to my rescue. It was very convenient of them. They answered the call, and so I want to start off with telling you a little bit about this year's Nobel Prize in Physics, which was awarded for the field that many of us here work in, the field of condensed matter physics, condensed matter theory in particular. So the Nobel Prize went to David Thouless, Mike Kosterlitz, and Duncan Haldane, all English theoretical physicists, all working in the United States. So in some sense, this prize, the fourth member of this prize is Margaret Thatcher, whose economic policies in England in the 70s and 80s contributed to the success of American science. Now, what did they get the Nobel Prize for? I don't want to spend my whole lecture discussing that, but let me just tell you a little bit about something that was part of their prize, and that's the phenomenon excuse me, of superfluidity. So here's a fluid. It's a fluid that you all know and love. Excuse me. It's a viscous fluid. It flows very, very slowly at room temperature. Here's a fluid that you don't know so well. Some of you do. Some of you in the audience are more expert on this than I am. And that's superfluid helium. Now, superfluid helium is an interesting thing, and perhaps for present purposes, all I want to say about it is that it's a fluid that in some sense flows without viscosity when you lower the temperature to very low temperatures. In this case, somewhere around 1 or 2 degrees above absolute zero. Take superfluid helium, cool it down there, and in a certain context, in a certain type of experiment, it flows without viscosity. And that's the sense in which we call it a superfluid. It has many other remarkable properties, but that's the thing that I want to focus on. So normal fluids have viscosity, superfluids don't have viscosity. And the question that Kosterlitz and Thouless were interested in, and the work that gave them the Nobel Prize, was what happens when you take a superfluid, but not in 3 dimensions, sitting in a jar or a dewar, but lying on a plane, just in 2 dimensions. Would just a single monolayer of atoms, of helium atoms, become a superfluid? Now, I don't want to go through the details of how they answered that question, but I want to tell you something about how they answered it. So how they answered it was to think about something very, very simple, something very, very abstract. They looked at what happens when you write down physics of what we'll call arrows on a plane. These are little arrows. They're meant to represent spin vectors or something like that, but really they're just arrows. And if you lay them down on a regular grid, you can see that you can make configurations of those arrows that look kind of like this one here. You can see that all the arrows go round and round in a circle, and you could in fact associate a particular number with the way in which they do that. So this flow configuration, this configuration of these arrows on the plane, you can think of it as being a little bit like the fluid velocity flowing in a kind of vortex. So, but that's just a kind of vortex configuration. And what they did was they studied the simplest possible model of the way that these arrows on a plane could interact with each other. Now, I'm not going to tell you what they did and how they did it, but what they ended up doing was predicting that indeed, in 2 dimensions, a superfluid can exist. And they predicted how that phase transition, the transition between the normal fluid, the regular flow of helium with viscosity, and the flow of helium without viscosity, how that can happen as a function of temperature. And here's the thing. This is experimental data for a quantity that you can measure. I'm not going to explain what it is. It's the superfluid density. This is the critical temperature, how low in temperature you need to go until the thing becomes a superfluid. And here you see the theory prediction, and here are the experimental data. In fact, some of these data were done here at the University of Illinois by Jack and Michelle. Now, why is this interesting? Why am I spending time to tell you about this? What's important about this for today's lecture is not what they discovered, but how they discovered it. Superfluids in 2 dimensions are really, really complicated. You would think you would have to understand something about the behavior of helium, the atoms of helium, the quantum mechanics of helium, all sorts of things like this. But it turns out you don't. They predicted how helium behaves just by looking at the simplest way that arrows on a plane could be described energetically. So the moral of this story is that if you want to understand some very complex phenomena, you can do so by looking at the simplest mathematical models that have the right symmetries, the right topology, various other things, right, simplest in a very technical sense. This is an amazing and surprising fact. It wasn't known 20 years ago, 30 years ago, and it certainly surprised and continues to surprise people by the power of these techniques. So in a sense, what I'm going to tell you then is about how one can try to apply the same kind of conceptual framework to biology. Now we have condensed matter theory represented here on our faculty. So here's some of the theoretical condensed matter physicists. They all work in very different areas of condensed matter physics, some to do with topology, some to do with things like avalanches and noise and things like this. So it's a very rich and broad phenomenon, and the University of Illinois is one of the premier institutions in the world where we study materials, study complex materials, study very exotic materials in this very sophisticated way. So how do you know theoretical physics when you see it? Okay, so here's a question, here's a quiz question. Okay, is this theoretical physics? How many people think yes? It looks pretty complicated, doesn't it? It's my blackboard, and I am a theoretical condensed matter physicist, so it probably is. But on the other hand, it's my blackboard in the Institute for Genomic Biology. So, is biology theoretical physics? Who knows? Okay? Well, the answer is that it is kind of theoretical physics, but it's not just theoretical physics. So let me explain that. Let's take a look at this part of this board. Is that theoretical physics? Okay, maybe. I mean, who the heck can tell, right? It's so complicated, and, of course you don't know. Well, the answer is nope. This isn't theoretical physics. It's mathematical physics. Okay? What I mean by mathematical physics, it's just a complicated calculation. It's like your maths homework problem, except on steroids. Okay? It's a very hard mathematical problem. It's not theoretical physics. How about this thing over here? Is this theoretical physics? Yep. This one is. This one is theoretical physics. You know why? Because this part isn't just a math homework problem. It involves a guess about how something behaves. A guess that you can't justify. A guess that comes from intuition, from putting pieces together, some kind of jigsaw puzzle, some kind of detective story applied to the physical universe to try to have some idea of how things work, and that is theoretical physics. It's a guess. So basically what I've shown you is there's mathematical physics, which is calculations, and there's theoretical physics, and theoretical physics is some simple guess, an educated guess, an insightful guess, an intuitive guess, a clever guess maybe, but still a guess. And then you do some mathematics to calculate what is the outcome of that, and then hopefully you can make an experimental prediction like Kosterlitz and Fowler's did, and therefore you can then test how good your guess is, and you can refine the guess and learn about the physical world in that way. So now when I want to talk about what theoretical physics can tell us about the evolution of early life, the question becomes, can simple models predict real biological phenomena like they do in condensed matter theory? So I want to try to convince you that the answer is yes if you ask the right questions and you ask those questions in the right way. Now the first problem that you come into when you are a theoretical physicist entering into biology, and by the way, I need to make a disclosure here. I hate biology. Okay, so I have no interest, in the mating habits of some fish or how many legs some kind of insect has or anything like that. I can't remember any of that stuff. It doesn't appeal to me. I can't calculate anything like that. So what is traditionally thought of as biology, I don't like and I don't understand, and I'm hopeless at it. Okay, but luckily biology is becoming more like physics. It's becoming much more quantitative. People are learning to think in different ways about biology, and that's happening right now. It's a really, really exciting time to be a physicist thinking about biological problems. Now, the problem with biology, as I sort of said at the beginning, one of the reasons why I hate it is that everything in biology has an exception. Okay, everything is different. How can there be some underlying principles? How can there be some rhyme or reason? How can you write down simple equations that you can put on a t-shirt and say this is biology, like you can with physics? When I first started doing biology, I had a great mentor, that's Carl Woese, a professor of microbiology, and when he summoned me to his lab and said, I want to talk with you, we talked for, first meeting we had was talking for about 4 hours, and I asked him about this. Everything in biology has an exception. What use am I going to be? And he said, no, it's not true. The people say that, but it's wrong. Everything in biology comes from evolution. Evolution is the underlying framework, the conceptual framework of biology, and there's no exceptions to that. Okay, and so we just have to understand evolution better. So that's what we worked on for 10 years on a daily basis. Now, this is our Institute for Universal Biology, which Peter mentioned to you. This is our logo. And I want to talk you through this logo because it's kind of interesting. It was put together not by a graphic artist. Well, the artist actually was the person who realized it, but the design of it came from me. Okay, so if it looks clunky and not very elegant, it's my fault. Okay, but I want to tell you about what the things mean on it because they will tell you about what I want to explain in the next half an hour or so. So this thing here is meant to represent the tree of life. What do I mean by the tree of life? Well, you all know that you have a family tree. You know that you have your parents, your grandparents, your great-grandparents, or you may have your children, their grandchildren, and so on. And everybody is descended from everybody else, and you can make a family tree. Well, you can do that not just for your family, but for all of life on Earth. And the first person who did that was Carl Rose. And when you do that, what you discover is this, that there are 3 branches to the tree of life, and it grows out like this. And this was the great discovery made here in 1977. Now, the question that we'll be talking about is, what's this thing over here? Because that tree of life was detected. It's not just somebody woke up one morning and said, isn't this a nice metaphor? It was measured, and it was measured by using DNA techniques that I'm going to tell you about in a simple way. But it's not, this thing here is not the beginning of life. The root of the tree of life is not the beginning of life. Life started from something, but it didn't start at the root of this tree. And what it started from, I think, is something like this. So this thing here is meant to represent not a tree, but a network. Okay? A network is the idea that life does not have a tree structure. There isn't a family tree. It's not a mandatory concept that you might otherwise expect. A tree is actually what we have now, but back in the day, life was organized like a network. And I'm going to show you why we think that, and the fact that this explains to us how life evolved so quickly. Now, Carl Rose was a wonderful scientist, but he invented horrible names for the things that he discovered or postulated. Okay? So he called this thing the progenote, and I might use that term in this talk, but it just means a phase of life which he didn't know what it was at that time, but we know today, and that's the network of life that I'll be telling you about. Now, this thing over here, this kind of looks like a spiral galaxy, right? Okay, you all know about spiral galaxies, and you might think that's a natural symbol to have because, hey, this is the Universal Biology Institute, and it's funded by NASA. So why not have a spiral galaxy? Well, it's not really a spiral galaxy. If you look at it, you can see that there's gaps in the spiral. Okay? In fact, those gaps are there to represent a genome. Okay? This is meant to be DNA. And it's not really a spiral galaxy at all. For me, it's a molecule that is more like a helix or a screw. Just like DNA is a double helix, this thing is meant to represent the screw nature, the chiral nature of a DNA helix molecule, or indeed any of the other molecules that you have in biology. They all have this, or many of them have this screw-like helical nature, and we call that chirality. All of these things, I think, and most people believe, are universal aspects of life. All of life is described by these types of phenomena, and that's why our institute is called the Universal Biology Institute, because it's not true that everything in biology has an exception. There are things that are common to all of biology. One of them is this chirality of the molecules, the fact that amino acids are left-handed, the fact that sugars are right-handed. And the other thing that is universal is the very thing that allows us to do genetic engineering, to take genes from one organism and plant them in another. And that is, every organism has the same code, the same genetic code. And I'm going to be telling you about that. Okay, so let me tell you then a little bit more about universal biology and make a connection to something that you all know and love, which is computers. So let's talk about the big question. So here's a headline that was on the various websites a few years ago, Mystery of the Missing Waves on Titan. Titan is a moon in the solar system. It has an ocean. In fact, it's methane, not water. And it should have waves on it because it has an atmosphere. And when there are winds, they make waves, right? Everybody knows that. It happens on Earth, it happens somewhere else. But we couldn't see those waves. And then later on, a few months later, relief. Cassini spies the wind-rippled waves on Titan. So why do I mention this? Well, the thing is this. We know that the waves on the oceans of Titan are missing because we have a general theory of fluid flow. We have a general theory of how liquids interact with wind and make waves. We can even tell you how high the waves are going to be and what their wavelength is going to be. Here's a headline that we haven't seen yet. Mystery of the Missing Life on Titan. Anybody ever seen that when they opened up their web browser? Don't think so. And here's one that I really want to see. Cassini spies the long-sought life on Titan. Have you ever seen that one? Well, sometimes you do see that, okay, but it's usually not correct. Now, why is this a joke? Why is that kind of headline not seen? Well, it's because you need to know that the life is missing. And we don't know if there's missing life because we don't have a theory that predicts the existence of life as a physical phenomenon. Normally, when we understand the world around us, we know why something exists. We know why fluids exist. We know why atoms exist. We can even work out exactly how many atoms there should be in the universe, what the number of elements, and how abundant they should be from studying stars. We know all of those things, but we don't have a theory that tells us that the phenomenon of life should exist. It's amazing, but we don't. So we need to develop such a theory if we want to understand whether life can exist on other planets. So the goal of our work, in some sense, is to come closer to a theory of life that can tell us whether or not we should expect life to exist, and if so, where and how. Now, we don't really have a real theory of universal biology today, apart from the things I'm going to mention in my talk. But we do have a theory of something different, something called universal computation. And this is a real theory, and it's useful. And I want to explain to you what it is, because that gives you an idea of what a theory of universal biology would look like if we were to get it in the future. So here's a computer, right? Just like my computer, okay? Some computer like that, made out of silicon chips and liquid crystals and so on. Here's another computer. Anybody here ever seen this computer? Anybody got one in their pocket? I didn't think so, right. Where have you seen this computer? You at the back.
It looks like the one they have next to Google.
It looks like the one next to Google, exactly, it is. It's in the Museum of Computation in Mountain View in California, that's right, it is. And it's also in the Science Museum in England. Why? Because it was built by Charles Babbage, Sir Charles Babbage, and it's a computer made out of cogwheels and springs and levers. Okay? He didn't finish it, but it is in fact just as much a computer as this one is. So this is kind of strange, isn't it? Here we have a computer on the left, glass, plastic, and silicon. On the right, cogwheels, springs, and levers, and I say they're the same. Why do I say that they're the same? Well, they both are realizations of an abstract concept, a mathematical concept. In fact, a concept developed by a pure mathematician, an English pure mathematician named Alan Turing, the famous Turing who actually also was a code breaker in the Second World War. And one of the things that he did was he developed the theory of computation, showing that mathematically it is possible to construct machines that can do any computation that can be envisaged. And he defined exactly how that is. And so today, these things are called Turing machines with a von Neumann architecture. So who's von Neumann? Well, von Neumann is this guy in the middle there. He's not a banker, he's a mathematician. And after the Second World War, what did von Neumann do? Well, they needed to build computers in order to design atomic weapons. And he took the mathematical framework, the pure mathematical framework from Alan Turing, and set about trying to figure out how you could design a computer built out of the technology of the day, thermionic valves, relays, and switches. And he succeeded in doing that. He was a mathematician, he designed these machines, he hired a bunch of electrical engineers, and they built them. They built them at the Institute for Advanced Study in Princeton, and they replicated that computer in several places around the United States, including here at Urbana-Champaign. And in fact, John von Neumann used to come and visit here regularly, and in fact gave lectures on these things that were published by the University of Illinois Press. So all of these things, they're all the same thing. The medium is not the message. These things are all examples of computation. Now, during the day, these computers at Princeton were solving very complicated calculations to do with designing hydrogen bombs. At night time, the engineers went home. And this guy, Niels Baricelli, who was a visitor to the Institute for Advanced Study, crept into the room where the computer was, and he did something very different. He programmed the computer to have computer viruses. Okay, you will know about computer viruses, unless you've got an Apple computer. Okay, so computer viruses, they replicate inside the computer, they breed, they interfere with things and so on. Well, he realized, he was interested in life, and he realized that you could make computer viruses that behaved kind of like living entities, and they could replicate inside a computer. They could compete, they could have wars, they could try to steal the resources of the computer from each other. And so what he did was he designed programs to do that, and these are some of his very first computer experiments, trying to simulate life, the simplest forms of life, viruses, inside a computer. Now, the way he did that is very interesting, and it says an important thing about evolution. In his world, in the computer that he was working on, these viruses could evolve. And when they evolved, they changed their own computer program. So the thing about these computer viruses is that their program was actually their data. And the data that they used was actually the program. So when they changed their data, they changed the way that the viruses functioned. So they evolved by changing their code. They changed the program that ran the viruses. It's a virus that directs its own evolution. And he discovered that they evolved and did many other things like this. His work is mostly forgotten today, but it really was foundational, and I will try to show you how we can use the same sort of techniques that he developed to try to understand the evolution of life. Now, what we have in biology is a different situation than we have in computer science. Because what we have in computer science is the idea, you start off with a mathematical theory, then you translate that mathematical theory into hardware. But we have a different problem in biology. Because in biology, we're not given the theory. What we're given is the hardware. We're given us. We're the hardware. Okay? So here's the analog. Here's a computer. Suppose I just gave you this computer, and I said, you can take it apart, smash two of them together if you like the wristwatches at the beginning. You can look at all the parts inside. You can do anything you want. Can you figure out that this computer is in fact doing the mathematical process that Turing described, the Turing machine with a von Neumann architecture? Could you do that just by looking at the computer? Well, I think you'd find it'd be a pretty hard job to go from all the silicon and chips and the liquid crystals and everything to the idea of a very mathematical description of computation. But that's what we have to do in biology. We have the hardware. We want the inverse problem. We want to know what is biology, what are the things that we are, the mathematical abstract theory, and what underlies how living systems work. And we need to understand that. Because if we understand that, then we can start to understand in more detail how life could exist on other planets. So if we're going to understand how life exists on other planets, we need to not assume that they use the same chemistry as us, that they use the same materials as us, the same atoms and so on. So we have to have a more general theory of life than just looking at our chemistry. And so that's really the goal of some of our research. Okay, so now I want to talk about an example of this. An example I want to talk about is the evolution of the genetic code. Okay, so here is a string of letters. Okay, anybody know what this thing is? It's what? DNA. Is it the genetic code? So who thinks it's the genetic code? So a few people. Okay. Well, it's not the genetic code. Okay. The newspapers always call it the genetic code, but as most of you said, it's the DNA. It's your genome. It's not the genetic code. Here's the genetic code. This is those A, C, G's, and T's. Those are the alphabet of the genetic code. They stand for these complicated biological molecules: adenine, thymine, guanine, cytosine. Doesn't matter what they're called or what they look like. They just have these symbols A, C, G, and T. And here's Francis Crick and Jim Watson with their model, their first model that described the structure of DNA as a double helix. Now, if I want to explain to you what the genetic code is and how it evolved, I need to tell you a little bit about what it actually is. So this is DNA. This is your DNA. This is the template by which your body functions, by which every cell in your body works. This is the master plan for an entire organism. Now, let's suppose we have something goes wrong with a sound system in this lecture room. Okay? And so we say, well, we need to fix it. So we need to call in the engineer to fix the sound system. So the engineer arrives, and we give her the whole plans for the entire University of Illinois at Urbana-Champaign. All the plumbing and the electrical circuits and the computer circuits and the buildings over this whole huge campus. And she's going to say, well, I don't need all of that. I just need the wiring diagram for this lecture hall. So she'll go and copy off a few pages that have the wiring diagram of this lecture hall, and then she'll fix the sound system using that. Biology does the same thing. You don't need to make a gene, you don't need to know all the other ones. So there's a machine called RNA polymerase, which goes along here. And this machine then reads off just the bits of the gene that you need, and that's called messenger RNA, also built from these 4 letters A, C, G, and T. For RNA, the T is replaced by another base called with the symbol U. Doesn't matter, still 4. Then you still haven't got proteins yet. U are your proteins. So you need to build your proteins, but your proteins are built from 20 amino acids. These are molecules, and you don't need to know what they are. You just need to know that there's 20 of them. So here you go. You have a machine that goes along this messenger RNA called a ribosome. This machine spits out the proteins. Now here's the thing. This ribosome makes proteins. It makes the proteins out of 20 amino acids. Those amino acids get strung together in some special order. The special order in which they're strung together is what makes the proteins work. Okay? If you get the wrong order, the protein isn't going to work. So how does this machine know which amino acid to string together in the right order to make any particular protein? Well, this is how it does it. It goes along here, it reads those bases, those AUGs and so on, in groups of 3. Why 3? You've got 20 amino acids, and you've got an alphabet of 4 letters. So how do you write a code that represents 20 amino acids out of 4 letters? Well, 4 only gives you 4. So suppose you say, let's read them in pairs. 4 times 4 is 16. 16 is less than 20. So if I read those letters in groups of 2, I will not have enough possibilities to read out 20 amino acids. So I need to go to the next one, read them in groups of 3. 4 times 4 times 4 is 64. That's bigger than 20. So if I read these nucleotides in groups of 3, I have enough combinatorial possibilities that I can represent a dictionary of 20 amino acids. And biology realized that, and so it goes along here, reads these things in groups of 3, and then spits out the right protein by bringing the right amino acid into the nascent protein. That's the ribosome. Now, what the genetic code is the map that's used by the ribosome to translate the message from triplets of 4 bases into the 20 amino acids of life. So it really is a code. It really is a code book. And it's an abstraction. It's a code book that tells you about all the complicated biology, all the stuff that I don't like about biology, all the chemistry, all the details, all of the things that I find hard to memorize. I don't need to know those because they can all be put into a map, into a code book, and the code book looks like this. And the people who figured out this code book, they also won the Nobel Prize. You read this like this. Here's the first letter. Here's the second letter. Here's the third letter. So if you wanted to read this thing here, you'd read this first column is U, the second column is U, and then if I look in this position, the third one being U as well, I would get this amino acid. This symbol stands for something called phenylalanine. And you would do the same thing for everywhere along here. So this is the code book that biology has evolved. It's called the canonical genetic code because every organism on the planet has the same genetic code. It's not like, well, there's one genetic code for humans, another one for honeybees, another one for rice, another one for jellyfish. There was a time when that was the case, but not now. Now everything has the same genetic code. And the question is why? How do we all have the same genetic code? So this is Carl Woese in characteristic pose on his desk, but not in characteristic pose on the front cover of the New York Times on November the 3rd, 1977. Carl Woese had just discovered a form of life that predates higher organisms. That's what the headline says. It's not really a form that predates higher organisms, but he had discovered a whole new form of life. Now, usually you hear in the news somebody discovers a new butterfly or a new lizard or something like that. Sometimes you hear that somebody discovered a new species. He discovered the highest possible classification of organisms, all on Earth, all living around us, and in fact even inside us, and nobody knew that they existed. What he discovered was the tree of life, and this is it. So this is bacteria, which you've heard about. These are archaea. Those are the things that Carl Woese discovered. And you can see over here are things called eukaryotes, and there's you. Okay, you are there. Everything else on this tree is a microbe. Okay? If you took all the humans off this planet, it would make no difference to life on Earth. Okay? Hopefully that experiment will not be done anytime soon. What we're interested in for the next few minutes is what's at the root of this tree. The tree has got 3 branches, but at the root of it, that's the organism, which was the organism from which we are all descended. It's called the last universal common ancestor. This work proved for the first time that all life on Earth is descended from the same creature, and all life on Earth is related. Now here's a puzzle. When they built that tree of life back in 1977, they realized something very strange. They realized that the organisms closest to the root of the tree of life were certain types of bacteria called cyanobacteria. And they knew even then that they were very ancient, at least 3.2 billion years old. Okay? Now the Earth is 4.6 billion years. These organisms, they said, were 3.2 billion years. Today, we know that those organisms actually were alive 3.8 billion years ago. So think about this. The Earth is 4.5 billion years. Life at least was detectable at 3.8 billion years ago. So that means that in less than 1 billion years, life was able to evolve organisms that were as complex as today's organisms, at least in the structure of their cell. And that was a surprising fact to them. They couldn't understand how it's possible that life could start off, go from nothing to the complexity of the modern cell in less than 1 billion years, and then after that, for the rest of the 3.8 billion years, nothing much happened. You know, we had a few dinosaurs, humans, things like this, but nothing very interesting, okay? So how did that happen? So what they said was, well, maybe this is George Fox and Carl Woese, this is how they did their famous work, very painstaking work using radioactive digests and films and so on. So, but what they felt was that evolution must have changed its character in the early days. Now today we think of evolution as happening by a sequence of mutations. That's what you've heard about. If you read the newspapers, you hear about mutations and things like this. Well, it turns out that that's really not how evolution works. Evolution works in a different way, and I want to sort of, in the last part of this talk, I want to just talk about the different way in which evolution really works. And they were right. Evolution was different back then. Now, the different mode of evolution that they really didn't think so much about at the time, but now we think is important, is horizontal gene transfer. So you have genes, and you transmit your genes to your children, or you get your genes from your parents. And then you transmit your genes to your children, and they transmit the genes to their children, your grandchildren, and so on and so forth. That's called vertical evolution. The genes flow down the tree of life. The genes flow to the organisms which are related to you. Suppose I wanted to do something different. I wanted to try to teach you some statistical mechanics. I wanted to try to teach you the mathematical physics, the theoretical physics, that those people at the beginning of this talk got the Nobel Prize for. I actually teach a class here on those mathematical and theoretical physics techniques. And they're quite complicated, and when the students go to those classes, they find them very hard to learn. Okay? Now, wouldn't it be nice if I could just say, take some genes out of my body, throw them out to you, you grab those genes, put them into your body, and then bingo, suddenly you know how to do those very complicated physics and mathematics calculations. Okay? That would make getting a PhD in physics a whole lot easier, right? Okay? Now you can't do that, right? Well, you can if you're a bacteria. Okay? Bacteria do this all the time. So what they do is they swap genes. They send genes out and they take genes in, and from those genes, once they take those genes into their cells, they can then express those genes and do something new that they didn't do before. It's called horizontal gene transfer because the gene is transferred from organism to organisms, even though they're not related. Now, the way they do that is through viruses, and they can come close together and transfer DNA, or they can even pick up DNA from the environment. Now, the question that people like me think about is this. Two bacteria doing this, that's very interesting, but what happens when they all do it? So when they all do it, you have really dramatic consequences. Here's one that I want to mention to you. This one is the spread of antibiotic resistance genes. Now, you may know, and you may have read in the newspapers, that the greatest health crisis facing us is not HIV or the Zika virus or cancer. Those are bad things. But it's not the biggest health problem we face today. The biggest health problem we face today is that the antibiotics that we use to cure bacterial infections, those don't work anymore. They're starting to fail. And they're starting to fail in dramatic ways. The bacteria have evolved resistance to it. It's an evolution effect. Now, you think of evolution as something like the scale of dinosaurs, millions and millions of years. That's the time scale in which evolution works. That's what people thought. But it's not true. Okay? The time scale for evolution is about 5 years. Globally, all the bacteria in the world, in a time scale of about 5 to 10 years, they can evolve resistance to the drugs that we use. Cancer tumors evolve resistance to chemotherapy very, very quickly in a few months. So biology works not necessarily by slow processes, but by very fast processes, and those processes occur, in some cases, by the horizontal transfer of genes. Okay, so I'm going to skip through some of this stuff because I don't have time to talk about it. So this is for a much longer talk. So what I want to then tell you about is how the genetic code evolved. So what we discovered was that the genetic code evolved rapidly at the beginning of life in the same way that the bacteria are evolving today their resistance to antibiotics. The same way using horizontal gene transfer. And here's how we did it. Here's how we figured this out. So what we did was we said, let's make a computer simulation of life. Let's make a computer simulation of organisms, just like the ones that Niels Baricelli did at the Institute for Advanced Study. We're going to make organisms that have a genetic code. They make proteins. They compete inside a computer. And the organisms can evolve. And they can also evolve the code in which they use, the code that translates their DNA into the proteins. So we make a computer simulation of that, and we then ask what happens when we do that. Now, what we found is something very interesting. So the thing I didn't tell you, the thing I had to skip over because I want to finish in 45 minutes, the thing that I didn't tell you about is this. Our genetic code is not just universal. It's not just the same genetic code for every organism on the planet. There's also something else that's really cool about it, something that was only discovered about 10 or 15 years ago. And that is, it's the best possible code you can get. Okay? It's a code that is the best possible one. Why is it the best possible one? Well, it minimizes errors. The thing about biology, the other thing about biology that everything that happens in biology is that it's very noisy. It's very fluctuating. All the signals are noisy. They're not very clean like in physics experiments. So what that means is this. When that ribosome machine goes along the messenger RNA and makes the protein, it makes lots of mistakes. When the DNA is replicated and transmitted from cell to cell, there's lots of mistakes in it. Those are the mutations. And yet, the genetic code functions in a way that is absolutely perfect. Just like when von Neumann was designing the first computers, he had to build a computer out of components that were always falling apart, always failing. Okay? That was the mathematical challenge. How do you make a machine that's guaranteed to get the right answer, even though the parts are faulty? That's what he designed. So biology solved this problem in a different way. It said, let's design a code so that if you get the wrong amino acid, the amino acid that you get, the wrong one, is still pretty close to the right one. And in fact, it designed it in such a way that it really is the best you can do in mitigating the effects of getting the wrong amino acid. So the special thing about the genetic code is it doesn't just give you the amino acids which you ask for, it gives you the amino acids which, if you get the wrong one, the wrong amino acid is still close enough to the correct one that the proteins will mostly still work. It's amazing, and we showed how that property evolved, and that's shown here. This is how good the genetic code is, measured in a way that I'm not going to explain. This is a function of time, and this is computer simulations of digital life living inside a computer. So here's what happens. Let's look at the red lights. So you start off here with your organisms, and you watch them evolve. And what you see happening is the genetic code gets better and better and better. Okay? The smaller number is better. As the genetic code gets better, say along this trajectory, suddenly it gets to this value, and then it stops evolving. It just stays the same. Nothing gets better about that genetic code. Now you run the experiment again. You get down to here, and then now you follow this line. Again, it doesn't change, but you get a different quality of code, a different ability to mitigate errors than you did the first time. You do it again and again and again, and you get all these red curves here. This is what happens if you use regular, vertical Darwinian evolution. This is the puzzle. Regular vertical Darwinian evolution is slow. You can't evolve a universal, optimal code just using the regular laws of evolution that we know and love today. Now you switch on horizontal gene transfer. Now you assume that the early life, before 3.8 billion years ago, was engaged in a marketplace, each organism selling and receiving genes from all the other organisms in some very chaotic fashion, very busy fashion. When you switch that effect on, then you see that you get the blue curve here. The organisms get better and better and better, and eventually evolve a genetic code that is optimal in the sense of minimizing errors. So what you learn from this is that horizontal gene transfer leads to optimality, that life prior to 3.8 billion years ago could only have evolved as rapidly as it has, could only have evolved a unique genetic code, and could only have evolved the best possible genetic code if we assume that life did not follow the usual rules of evolution that we're used to today, but instead was dominated by this network of genes switching and swapping between the different organisms. We can do the same calculation and just check that when you do that simulation, the life develops a unique genetic code. So what we learn is horizontal gene transfer, this network of life, leads to universality and optimality. Today, we have the tree of life. Before 3.8 billion years ago, we had a network of life. So what do I learn from that? Here's what I've just told you. Here's Carl Rose's tree of life, the three domains: bacteria, archaea, and eukaryotes. Here's our cloud of ignorance surrounding about the origin and the evolution of life. So what I've shown you is that life started with a state which was kind of collective. It was kind of a network. But it started off with different organisms with different genetic codes. One for this class of organisms, one for this class, one for this class. And those organisms competed and eventually created a state where there's only one genetic code, and it's the best possible genetic code. Now we have a very interesting situation. You have organisms that have the same genetic code. They can all swap genes now between everything. And so what happens is you get a communal state with an explosive growth and innovation sharing. If one organism invents a new gene, that gene can be shared throughout all of life. The analog of this is something that happened in 1992 and 1993. Can anybody guess what it is? What happened in 1992 and 1993? What happened was the birth of the internet. Okay, before that time, we had a technical way for computers to transfer information, but at that time, a new protocol was invented, Hypertext Transfer Protocol, HTTP. You all know those letters. Every website you go to has those letters. And that protocol was invented around then, and suddenly every computer in the world could talk to each other. And then you have an explosive growth in the development of the internet. It's called an internet because it's a network. And that networks grow fast, much faster than individuals do. And so there was an explosive growth of innovation that led to the internet boom and the internet that we have today. So that's what biology did too, but it did it 4 billion years ago. Eventually, life became so complex that method doesn't work, and then you made a transition to the modern era where we now have just the vertical Darwinian evolution that we know and love. You get the transition to the tree. All right, so let me end. So, what can theoretical physics tell us about the evolution of early life? Can simple models predict real biological phenomena like we do in condensed matter theory? So my answer is yes, they can, if you ask the right questions in the right way. So here's my take-home messages. So theoretical physics starts with a simple guess, and from that you can calculate and you make comparisons with experiments. Simple models, very simple mathematical models, can describe very complicated phenomena. And we should try and understand the universal features of systems as complicated as living systems, because there's a chance that the methods of theoretical physics can be successful, and we can understand such complex things as the genetic code, or the chirality, the helical nature of biological molecules, from very simple arguments. So that was what I wanted to tell you about, and I'll be happy to take any of your questions.
So if there's horizontal gene transfer, do we still have a tree of life?
So the question is, if there is horizontal gene transfer, do we still have a tree of life? So that's an excellent question. When people discovered horizontal gene transfer and started thinking about it, they asked exactly this question. Now, the thing is this. When we build the tree of life, that tree refers to the genes that make the translational machinery, the core operating system of the cell, the thing that reads the DNA. So when we say you're related to some other organism, or that organism is in the same species as you, what we mean is you have the same DNA for the machinery that does translation, that makes your proteins. And so when we build the tree of life, we talk about that machinery. Now what I'm talking about is horizontal gene transfer of those genes, those very genes that define species. So when you have a network, you don't have species. Now, today we do have horizontal gene transfer, but it's not of the essential genes. It's not of the genes that define a species. They're the genes for things that aren't very essential, like antibiotic resistance, for example. Those genes are quite easy to come by. So you can have, for the core genes, the genes that define the organism and the species in the cell, those follow a tree, but other genes can be horizontally transferred under some situations, and so there's no contradiction between those two things. But it's a very important question, and to this day, people ask about, does horizontal gene transfer destroy the idea of the tree of life? It doesn't, because you're talking about different categories of genes. So did I answer your question? Would you have a follow-up?
I'm just wondering if you could imagine species fusing.
You could imagine that species fuse, yes. Some people think that organisms such as us, eukaryotes, came from the fusion of archaea and bacteria and so on, and we certainly know that some parts of our cells did come from some organism eating another one and engulfing it. So that definitely happens. Yeah, exactly right. Yes? How do they decide which genes to swap? Is the question, is that right? Yeah. Yes. So they don't decide, okay? They just swap all of them. And then what happens is, some of the genes that get swapped are useless, or you only swap to half the gene, so it doesn't do anything. But if you do happen to swap a good gene and it helps you, then you can make more babies, and then you become more successful. So what we end up seeing is only the genes that get swapped that were actually useful. But actually all of these genes are being swapped in some crazy way, and most of the time it doesn't do anything useful, but some of the time it does do something useful, and those are the times that count. Okay? Did I answer your question? Okay. Yeah.
I just wondered, does this work have implications for the search for life on other planets?
It does. It does have implications for the search for life on other planets, because I think that if we want to know whether life exists on other planets, one way we're going to answer that is by knowing that life should exist. We understand the theoretical reasons for life to exist. The other implication it has is that we know that we understand now how life can evolve rapidly. It's not the case that you can say, I have a planet and it's only 3 billion years old, so how could they have intelligent life? Well, heck, 1 billion years is enough. Probably 500 million years is enough. So the fact that we understand how life evolves so quickly makes it much more likely that we're going to be able to find environments on other planets where life will evolve. And I think we don't need to look very far. I think we'll find life on Mars. I think we'll find life on Enceladus. We'll find life on Europa. I think many of these moons and planets in the solar system, they have oceans of liquid water. They have all the conditions necessary for life, even as we know it. So I don't think we need to look very far. But that's not a scientific statement. It's a question of belief at this point.
Can physics and chemistry begin to help us understand how things began to self-assemble in the primordial soup when individual proteins were floating around or membranes began to form?
Yes. So the question is, can physics and chemistry help us understand how life began to assemble early on? So I haven't really talked about that because as you said in your question, it's a very good question, it's not just physics, it's also chemistry. You also have to understand what are the molecules and how they come together and how they form aggregates. And so the answer is, yes, people are trying to do that. And we're trying to do that here. We're trying to find the environments in which life could have assembled, where molecules could have come together and started to form membranes that would encapsulate the contents, which would form nucleic acids and so on. So people are trying to understand exactly those things. But I didn't talk about those things in my talk because you require a greater level of detail than what I was talking about. And we don't really have a good understanding of those processes, how those things got started. So that's still a very controversial subject right now.
What was it that Carl Rose found to spread the tree of life to the archaea?
So the question was, what did Carl Rose discover that spread the tree of life to the archaea? What he did was he looked at molecular sequences. That was his thing. People before that, if you wanted to classify an organism, you'd look at its shape or its color or how many legs it has or something like this. And he said, well, I'm a physicist. Carl Rose was a physicist. So he didn't really care about all of that physiology. So he said, let me just look at the molecules that code, that are the DNA, for the ribosome, this machine that is present in every cell. And then he started looking at those sequences and seeing which ones were similar to others, and that's how he could group organisms. So what he was expecting was to find two groups. He was expecting to find organisms that were called prokaryotes, which don't have a nucleus, and organisms that do have a nucleus. That's what he was expecting to find. And that's what he did find when he started doing his work. But pretty quickly, Ralph Wolf gave him some organisms that he was working on here at the University of Illinois. And when Rose looked at the molecular sequences characteristic for those organisms, they didn't fit into the patterns that he'd seen for all the other organisms. In fact, what he did was he ran into Wolf's office after he sequenced them, and he said, Ralph, these organisms aren't even bacteria. He couldn't believe what he was seeing because it was so unexpected. And then they did more and more of them, and they discovered that these things do exist. And then now they had the idea that there was this new class of organisms. They looked at them a bit more closely. They discovered that they're cell membranes were somewhat different from the bacterial ones, and they discovered many other differences between bacteria and these archaea. So even when you look at them under a microscope, they just look like round blobby things. Okay? You know the saying, don't judge a book by its cover. Okay? Well, the same thing is true with biology. What was discovered was that they look the same, but they're not. Their evolutionary history is completely different. And that's what he discovered. So did I answer your question? Yeah. Thank you.
I was just wondering sort of like what role epigenetics plays in this story. So the idea that environmental exposures that pregnant women are exposed to can impact the kind of development of the children at a genetic level. It seems like it's a bit random. Can you use physics to sort of like model that, or how do you fit that into this story?
So the question is a very interesting question. The question was, where does epigenetics fit into this? Where do environmental influences affect the DNA and affect your offspring and so on? And so it's a great. Did I say your question correctly? Yeah. So the answer is it's a really, really important question. People only now are just realizing that that you can change the state, that just looking at your DNA sequence isn't enough. There's a hidden code, a code that actually we still don't fully understand. And that code is when you do something in the environment. For example, even if you do something as simple as jumping up and down and exercising for half an hour, you will change some of the state of your DNA in your cells. And that will change the way that they behave, and it will certainly change the way that they evolve, because it will change the way that you function in your environment. So you're absolutely right. Epigenetics, this extra code, this extra bit of information that tells you about whether genes are expressed or not expressed, that does affect evolution. And actually, I'm quite sure that the methods of physics that I talked about can make simple models of how that works. And we're actually, in my group, we're trying to understand how environmental influences affect evolution. But we don't have a really good answer at this point yet. Maybe if I come back in 5 years' time, I can give you a better answer to your question. So thank you, that's a really great question. Yeah.
So how does viral life relate to the tree of life?
The question is, how do viruses relate to the tree of life? Yeah, so the tree of life was constructed for organisms that have cells. So the statement is all life on Earth has cells, right? Well, it's not true, as you point out. Viruses aren't really cells, and they don't have ribosomes, because what they do when a virus goes into a cell, it hijacks the cell's machinery to make baby viruses. So it doesn't carry its own machinery. It's a kind of parasite. So this idea of the tree of life, and can you use it to classify viruses? The answer is no. The viruses aren't on the tree of life. We don't have a good way to classify viruses in the same way that we can classify cellular life. So if you have an idea, please tell me, because we don't know. All we know is that viruses are extremely important in understanding evolution and understanding ecology today. And we use mathematical models to describe how viruses interact with bacteria and affect the way that the bacteria evolve and affect the way that the environments evolve. So we do that in my own research group, but we don't have a way to classify viruses in anything more than the superficial way. Yeah.
Mammals as individual organisms with vertical gene transfer, but we actually are also cities for 1 trillion microorganisms in close proximity.
Yes.
And so do you ever measure, like for individuals, a mammal can live 100 years, whether horizontal gene transfer within a mammal affects both the mammal, but also the rate at which their microbiome is evolving?
Right, so the question was, when you think of us as humans, we are basically a zoo. And inside the zoo are 1 trillion bacteria and archaea that live inside our gut. And the question was, is there horizontal gene transfer going on inside us, and does it affect us as well? So the answer is yes. Okay, it's a very good question. So the bacteria in us are horizontally transferring genes, and in fact, that does affect our health. It affects our ability to withstand bacterial infections. It withstands our ability to have an immune system that is robust. So the evolution of the microbiome inside our bodies through horizontal gene transfer does affect the host organism. But there's something more. We know that in some situations, the microbes that live within us can actually transfer their genes to the host. Now, we don't know if it's true to humans. I don't know, that may be true, but I don't know it to be true. But we do know it to be true for things like fruit flies, Drosophila. So fruit flies have bacteria that live within them, Wolbachia. And what we know is that Wolbachia have horizontally transferred genes into the genome of the fruit fly. In fact, they've transferred the whole genome of the bacteria into the genome of the fruit fly. And we've now uncovered many examples where the genes get horizontally transferred between bacteria and plants, or between animals or insects and so on. So that was only known, that wasn't known 10 years or so ago. And it's still controversial, and people are trying to understand the extent to which it occurs and what are the influences of that. But I'm sure we're in for a lot of surprises. I'm sure we're going to find that it does affect the evolution, and so it's a story that's still unfolding. So thank you. Good question. Yes? How did life begin? I don't know. But what I do know is that if life began, I know where I think life began, but if life began once, could it begin twice? Right? If something made life start, maybe it could happen again, right? And maybe it happens again and again and again. So let's suppose, I happen to think that one place where life began was right underneath the ocean, in volcanic regions where there are what are called hydrothermal vents, regions where there's hot rocks and lots of minerals that come together, and there's a huge, if you like, soup cauldron of chemicals that all get mixed up, and people think, for various reasons, that out of that life emerged. And some people have some ideas about how, but nobody really knows. Now, if that's happened, say, 4 billion years ago, it could still be going on today. We could still be finding life being created right now. So I think your question is one that we could answer by actually trying to find the environments where life started and looking to see if life is still being created there. But there's a problem. And the problem is this. Suppose we could see that kind of life. How would we know that it really was a life? Because how do we recognize life right now? We look to see if it has DNA. If it has DNA, we know that thing is living. But early organisms didn't have DNA, for sure. DNA didn't always exist. It was created by life. So if we want to look for how life started, we can go to the places where life started, and most likely it is still being created there. But those primitive forms of life won't have DNA. So we have no way to detect that life. We don't know how we would recognize it as living. So you think about that. Come up with a good answer. If we look at a rock down at the bottom of the ocean, and it's got all the conditions for making life, but there's no fishes or anything like that, or shrimps or worms or anything like that. There's just some very primitive life forms before there were even cells, before there were species. Well, how would we know how would we recognize that kind of life? So we don't know. People are very interested in answering that question, because if we want to go to Mars and look for life there, we have to ask ourselves the same question. If we can't detect simple life on Earth, how can we detect simple life on Mars or some other planet? So your question is a great question. I don't know the answer. I wish I did, but it's a very important question.
So the systems are complicated, but we want simple models. So when we validate the models, how can we remove the effect of the confounders? What's the methodology of the principles?
So the question was, the phenomena are very complex and we want to use simple models, so how do we remove the effects, all the complicated detail effects? So the answer is that you can't. Okay? So you have to ask questions in just the right way. So there's some questions that have simple answers and some that don't. Let me give you an example, something very simple. Let's go back to the work that won those people the Nobel Prize this week. Okay? So they made this beautiful, very profound theory of superfluids in 2 dimensions. And they predicted that at low temperatures how superfluids could form. But suppose you ask them, how low in temperature do we need to go before the fluid becomes a superfluid? Their theory doesn't explain that. We can't answer that. Because that really does depend on the atoms, and it depends on what surface it is, and many other things like this. So some aspects of the phenomena don't depend on the details, and some do. And what physics is successful because we mostly ask questions that don't depend on the details, like those guys who won the Nobel Prize. Okay? They knew how to ask the right questions that didn't require us to know all those details, the confounding factors that you talked about. And so the moral of the story is it's not just, well, you're lucky, just make a simple model and it'll work. It's not like that at all. When you make a simple model, you have to be very, very careful to ask the questions in just the right way. So for example, my work on the genetic code, okay, I can describe how the genetic code would have evolved. But if you ask me, why are there 20 amino acids, not 18 or 25? I can't answer that. That's a chemistry question. Okay? Some people did try to answer that question, but I don't think it's a sort of question that the tools of theoretical physics are suited for. Okay? So you have to use different techniques to answer those kinds of questions. And those questions will not have simple answers. The answers will depend on the history and chemical details and very complicated things like that. Okay? Yeah.
There were 64 possibilities with those.
Yeah, 4 times 4 times 4. Yeah.
It only does 20.
Yes.
20
20 amino acids. Yes.
Amino acids. So what happens with the other 44 possibilities?
Right. So the question is this: I've got 4 times 4 times 4. That's the triplets of those of nucleotide bases. So in principle, they could code for 64 amino acids, but for some reason, biology only used 20. So what about all the other ones? Okay. So if you looked at the table, I don't know if I can quickly get to it. So let me see if I can. Oops. There was one there. There it is. Okay. So if you look at this table, what you see is that this one here, for example, valine, this is G, and then U, that's over here, and then any of U, C, A, and G will still give you valine. Okay? So this code is not a one-to-one code, it's many-to-one. So that's the answer. Okay? Yeah.
So if we know the good genomic code, can we introduce into the commonly known bacteria, and then commonly known bacteria, even though they do the horizontal gene transfer, can we treat that to treat antibiotic resistant gene problem?
So the question was, let me see if I can understand your question. I think what you're asking is, could I change the genetic code of bacteria so that they are more susceptible to antibiotics by using the same genetic code? So yeah, that's an interesting question. Can we find ways to reprogram bacteria so that they can be killed? So I've thought about that, and I think you can. I think there are some situations where you could deliver genes to the bacteria exploiting the genetic code to kill them. Now, I wrote a proposal to do something like this. The proposal was, one of the ways that you get infections is if you go to hospital and you have some injections or some needles in you and things like this. The bacteria, you think of bacteria as little blobby things that are floating around. But what they do is they accumulate and form a kind of slime. That slime, you can feel the slime on your teeth. Okay? That's called a biofilm. It's a collective of a whole bunch of bacteria. It's kind of like a city of bacteria. So they form those things, and then once they're very close together, they can horizontally transfer genes very easily and become resistant. So I thought that one could exploit that and exploit the genetic code to then transmit poisons, some toxins in some way throughout the biofilm and then kill the bacteria, because the biofilms are the very bad things for hospital-borne infections. So I think you can do that. The thing is that you need funding to do that, and when I wrote a proposal to the National Institutes of Health to do this, my proposal was reviewed by a panel that consisted of child psychologists and chemists and people like this, and so they had no idea what I was talking about in this proposal, and so it didn't get funded. Okay, but I think, and we had a good idea of how to do this, and in fact a way to actually stop the evolution of bacteria, a new way to treat them so that they don't evolve the resistance. But it's hard to explain it to people. I mean, the fundamental thing is that living things are not like engineered things. Engineered things, you just take out a part, replace it, and it works again. Biological systems, when you interfere with them, they respond in a completely different way. They do some new function that you never saw before, and that's how they root around the problem and fix things themselves, and so then they start doing something completely different. Living systems do that. Even systems that are kind of living, like financial markets. Okay? Financial markets are made of all sorts of agents, people and companies, all trading and doing some very, very complex activity. And if you want to make a financial market do something, you want to fix the economy, you want to put on some new taxes or some new regulations or something like that. So the Federal Reserve tries to do that, but it fails. Almost always it fails, because the market then evolves some new kind of instrument or some new technique or some new tax dodge or something like this, and they figure out a way to not be hemmed in by the regulations that you try to impose. So living systems that can respond to their environment in ways by creating novelty, do things that you can't anticipate and can't understand. And we don't have a very good way to think about how to model such systems. So going back to your question, yes, I think one can exploit the genetic code to try to manipulate biology, but if we're going to do it, we have to do it in a way that doesn't just treat organisms as machines. We have to recognize that they are living and that they can evolve.
Perhaps one more question.
Yeah.
So I saw in the tree of life, it started with like a network, and then it came together when everything decided on a code, and then it kind of branched out from there. So are there any models or ideas for how the spreading into an actual tree occurs?
Yes. So the question was, if life started with a network and ended with a tree, how did the tree emerge from the network? Is that your question?
What was the driving force to cause a split in the tree is my question.
The question is what is the driving force that creates a split in the tree? Why are there 3 branches? Why not 1? Okay? And so I don't know the answer to that question in detail. I've asked the same question in a slightly different way. My way of answering the question is, why are there 3 branches to the tree of life, not 10 or 100 or 1,000,000? Okay? Now, the reason I think that the tree splits in this way is because each branch is a specialist in some particular type of living function. So one branch might be very good at replication. One branch might have concentrated more on metabolism and things like this. So I think the reason for the different branches is that because when this network sort of went unstable and produced the trees, the different branches were specialists in some particular function of living systems in some broad way. Now, I can't really prove that. We don't know how to calculate that. And when we look at these organisms in detail and look at their biological characteristics, you can kind of convince yourself that that is what happened. But it's a just so story, okay? And I didn't want, and Carl and I didn't want to publish that because we wanted to have a mathematical model that said this should occur in this way, there should be branching like this. It's something we're still working on, okay? But I think that's likely to be the explanation. At least that's what we think is going on. That's our guess. I told you at the beginning that theoretical physics was a guess followed by a calculation. I have a guess, but I don't have a calculation. Okay.
Okay, that's probably a pretty good place to stop. So let's
Okay.
thank Nigel once again.